Hello, everyone. First, I'd like to thank A-Star Research Support Center for organizing and supporting this webinar. And thank you all for joining us in this episode of Connecting the Dots with Azenta, Overcoming Challenges in CAR-T Therapy with Single-RON. And we are pleased to have Dr. Maxine Lam from IMCB Singapore to present her work on CAR-T therapy. I'm Joel, your Azenta Technical Application Specialist for APAC. And I'd like to give a quick introduction of Azenta. Azenta Life Sciences is a MNC headquartered in the US with manufacturing, laboratory, and biobanking services and facilities in US, UK, Germany, China, Japan, and Singapore. Azenta brands include Brooks, Biosigin, Fortitude, and GenWis, and we cover a wide range of products and services. Of relevance to today's edition is Azenta's multiomics and synthesis services under our GenWis brand. We provide a wide range of next-generation sequencing and gene and oligosynthesis solutions. And here are some recently published peer-reviewed studies that have used Azenta's NGS services. We are platform agnostic and provide sequencing services using the four main platforms: Illumina, PacBio, MGI, and ONT. I will now hand over the time to Dr. Sunjata from Single-RON.
Thanks, Joel, and thanks, RSC, for this. So as mentioned, I'll do a quick intro on what we can provide as Single-RON, and then introduce Dr. Lam at the top. So Single-RON, we are able to provide sample-to-answer single-cell solutions, and by which I mean we have products and kits that span the whole single-cell workflow, from tissue preservation, dissociation, library prep, and data analysis, including customized data analysis. And what we can also provide is single-cell analysis services, by which I mean if you're not interested in doing your own library prep and workflow, we're a one-stop solution for this. We can consult on your project, bring in your samples to the entire single-cell workflow, and provide you with data analysis, including customized data analysis. We've worked with many different sample types and tissues, including in vitro models that you will hear about in the upcoming talk by Dr. Lam, and do contact us for any of your single cell requirements, and we can help you through that. So without further ado, I will introduce Dr. Maxine Lam. So Dr. Maxine Lam earned her doctorate in developmental and stem cell biology from MRC LMC in London as a Wellcome Trust and A-Star PhD fellow. She was a recipient of the Career Development Fund by A-Star, as part of which she developed a novel in vitro model of glioblastoma that incorporates a blood-brain barrier. And this mimics the complex tumor immune microenvironment. In her presentation, Dr. Lam will now share insights from investigating the efficacy of CAR-T therapy against GBM using in vitro models. Dr. Maxine Lam, please.
Hi, okay, great. Thank you so much for the kind introduction. Is that right? No?
Yeah, correct.
Great, okay. So yeah, moving ahead. So as you know, glioblastoma is a pretty deadly disease. It's the most common brain or CNS malignancy. Typical treatment consists of maximally safe surgery, which is tricky because obviously the brain is very important, so you can't just chop it in half, followed by radiotherapy. And then since 2005, concurrent temozolomide. Unfortunately, recurrence is inevitable. And that does mean that the median survival for GBM is around 15 months, despite improvements in surgical and radiotherapy techniques, and essentially one chemotherapeutic option. So GBM is notoriously difficult to treat for many reasons, but I think I'll just begin with the structure of it. So GBM is characterized by a dense necrotic core, which is also hypoxic, and it's surrounded by invasive cells. And probably due to its dense nutrient-sparse tumor core, tumor tends to be pro-angiogenic, and as a result, GBM tumors tend to be highly vascularized. The invading cells in the periphery actually use this vasculature to migrate along, and also they invade in the parenchymal space, also sometimes along axonal tracts. So this total kind of organization of the tumor makes it very hard to resect surgically. So It's been known for quite a while that the organization of tumor cells from the core to the periphery affects a wide range of tumor biology, such as metabolism, proliferation, gene expression, signaling. But it can also be a physical barrier to drug and cell therapy access because it's so just solid. And the brain's vasculature as well is involved in tumor behavior and drug delivery. So I've mentioned that the brain's vasculature can be hijacked by tumor cells for nutrients and also for invasion. And in particular, the brain vasculature is highly specialized because it has these parasites wrapping around the basal sides of the endothelial cells with astrocytic amphiid and interneurons connecting the vessels that all together basically form what is known as the blood-brain barrier, or the BBB, which you hear me use that phrase, that term a lot. So and the BBB's permeability is highly controlled because it's evolved to prevent toxins from entering your brain, to protect your brain. But this also means that most chemotherapies are unable to cross the BBB because of this very tight permeability. However, the BBB has sometimes been known to be disrupted in GBM, but rather than increasing access to drugs to the tumor, it can sometimes lead the therapy to leak out at non-tumor regions. And then this leaky BBB can actually lead to edemas and hemorrhaging in patients, which are, of course, as you can imagine, incredibly debilitating. So given the roles of tumor heterogeneity and the BBB and GBM tumor biology and drug resistance, we felt that these features were very important to recapitulate if we wanted to study this disease in vitro. So we started with modeling the solid tumor itself. So for this, we used immortalized commercial GBM cell lines, such as U87, which you can see in the micrograph there. Or we also sometimes use patient-derived GBM cell lines. For today's work, I'm not going to be talking about any of our patient-derived cell lines, but you're welcome to ask questions at the end. So key to forming this is that we have to make spheroids of at least 500 microns in diameter because previous research has basically shown that, this is the minimum size required to start seeing changes in the tumor biology, especially hypoxia. And then for the BBB, what we do is we resuspend a mix of primary brain endothelial cells, brain parasites, and astrocytes in a hydrogel, in a fibrin gel. And this took about a year and a bit to optimize, and we've just submitted a Nature protocol paper for this. But essentially, what we then do is just we resuspend all of them together, and we inject it into a commercial microfluidic device, which you can buy from in biotech. And what this microfluidic device has, as you can see, is that it's got like a central channel here. Actually, can you guys see my cursor? I don't think so. Hang on. Let me use the laser pointer. Central channel here, where the tumor is, surrounded by the cells, and is flanked by a media channel on top and bottom that allows for basically perfusion without disturbing the gel in the middle. And over a period of 5 to 7 days, what happens is that the cell types essentially self-organize under the right conditions and form a perfusible network of vasculature. And so here I have one such device that's been fixed and stained, just to show you kind of what it looks like. And in green, we have the tumor cells with the dense tumor core. And you can see the invasive front here. In yellow, you have the vasculature. In pink, you have the astrocytes. And in blue, you have the parasites. And I put this next to the cartoon to show you kind of how strikingly similar the tumor looks compared to the cartoon. And I think this just highlights kind of the importance of kind of doing biology, cancer biology in 3D with the right conditions, because then you can start to really observe, real biological phenomena. So another feature that's very vital to replicate was the selective permeability of the BBB. And so you can measure this by basically perfusing fluorescent dextrans of different molecular weights and then measuring the flux into the extravascular space. So we were able to observe restricted permeability for high molecular weight dextrans, such as in here, you can see 70 kilodalton. You can see here, I have no tumor and tumor. You can see in orange, basically, that the vessels are completely perfusible. And then you can do the same with the lower molecular weight, which is 3 kilodalton. And then you just measure. And what we see is that there's very tight permeability for 70 kilodalton and 10 kilodalton. And then obviously the permeability is higher for 3 kilodalton because they're smaller and there's a bit more diffusion happening. And then what we notice is that when you actually have a tumor present in the BBB, this very tight regulation, especially to the 10 kilodalton dextrans, gets lost and it becomes a lot more linear, which basically means that you have a kind of a permeability that's just purely dependent on the molecular weight. So this also is kind of a phenomena that's observed in clinical patients where there's a loss of permeability control in the BBB when the tumor is present. So, but most importantly, how does this compare to what happens in an animal or a patient? And so when we compare actually the values that we get in our model, it's quite encouraging that that we see essentially that they're in the same range as that was values that are obtained in the rat brain for all the different molecular weights. So this basically gives us confidence that when we do in vitro studies for BBB permeability or extravasation, we're actually seeing something that is like relevant to what would happen in a patient or an animal. So now that we have a nice model for the BBB and the GBM, we can start thinking about how to use the model to study tumor microenvironment and its interactions with therapy. So the TME is particularly important to consider when developing immunotherapy such as CAR T cell therapy, because CAR T cells face multiple obstacles inside the body. First of which is extravasation, where they have to leave circulation and enter the target tissue. And then after extravasation, they enter the tissue space, which is likely to be immunosuppressive because it's been conditioned by the tumor over years, months to evade the immune system. And if they remain functional and are able to target the tumor, there's also the issue of antigen loss in the tumor, where some cells in the tumor actually downregulate the target antigen to evade T cell cytotoxicity. Besides this, there are undesirable side effects of CAR T cell therapy, such as off-tumor toxicity, where non-tumor cells get targeted by the CAR T. And there's also a phenomenon known as cytokine release syndrome, where the CAR T therapy essentially triggers the immune system to overproduce cytokines, leading to a hyperinflamed state that is actually really detrimental to the patient. So, but despite these risks, immunotherapy and CAR T therapy in particular remain a very promising option for treating GBM, because I think the most notable case that gets kind of cited is the one that was recorded by a clinical trial by Brown et al. Performed at the City of Hope Hospital in the US, where they observed a complete remission in a patient using an IL-13 Rα2 targeting CAR T. However, eventually this patient's tumor recurred, and they found possible antigen loss to be responsible for this recurrence. But I mean, overall, there's a lot about the brain's tumor microenvironment that is unknown, but studying this is very difficult because you need patient material, which is obviously not always accessible, or you need to do very complicated animal experiments. So we therefore thought that our microphysiological model could actually be very useful in this case to study the tumor microenvironment of GBM and how it affects CAR T cell therapy. Given it's reproducible, it's highly controlled, it's easy to use, and it's able to recreate essentially many of the physiological obstacles found in patients that I just listed, such as the BBB and the immunosuppressive TME. So I'm going to skip ahead quite a bit because I really want to get to the single cell data soon, given the nature of this seminar. But essentially what we did was fairly straightforward. We generated IL-13 Rα2 expressing U87 cells. We cultured them like I described with a BBB. And then when the BBB was perfusable, we added fluorescently labeled IL-13 CAR T to target the tumor, and which is basically what you're seeing on the left side of the slides right now. So you can see that because also the device is optically clear, we can carry out live microscopy and track essentially the same tumor over time. So you can see that on day one, you have the tumor in green and the CAR T cells in orange. And you can see that the CAR T cells are basically very good at targeting the tumor cells pretty much immediately. And by day 4 and essentially day 10, there's the CAR T have done their job. They've kind of died as a result. And also the U87 cells have also been reduced quite a bit. But you'll notice that actually the ones that were targeted by the T cells were actually the infiltrating invasive T cell, tumor cells. And the tumor core actually appears to be resistant. Sorry. And so, for comparison, in a similar microtumor, here we have one that has not been treated by CAR T. So sorry, 1 second. Sorry, there's no lock on this door. So yeah, so we have a tumor tissue that's been not targeted by CAR T, and you can see that clearly there's all the invasive infiltrating tumor cells, whereas here there is only the core left. But we were very curious as to why the tumor core appeared to be resistant to CAR T cells. And, so we kind of dug a bit deeper into it. And here you can see a fixed and stained confocal image of the same microtumor after CAR T treatment. And now in blue, you can see the vessels. I should have mentioned before that here, all this black space here is not actually empty. It's full of BBB cells. They're just not labeled, so you can't see them in the live microscopy. But here in the fixed stainings, you can see them. So in blue now, you can see the vessels here, and you can see very clearly that the tumor core, which is here in green, is still present, and the T cells in yellow are struggling to extravasate, and a lot of them are actually trapped within the tumor, the vascular lumen. And in purple, you can see a marker for apoptosis, and so a lot of them are also apoptotic, so they're starting to die off by day 10, and they're trapped within the lumen. So it seems like lots of the different cells. So immediately we have some clues that the tumor microenvironment may be affecting CAR T cell therapy. But to understand what the different cell types are doing in the TME, we are using multiple approaches. So what we can do is actually we can remove the microtumors from the devices, and then we can digest them to single cells, or we can section them, we can embed them and section them like regular tissue. And then we can do all your single cell assays or your spatial assays to understand the tumor microenvironment. However, in the interest of time, also because of for today's talk, I'm just going to be talking about the single cell RNA-seq that we did. So just a quick overview of the entire experimental outline. We basically, even though I've only showed you images from the tumor plus BBB microtumors, for the experiment, we obviously had controls. So we compared the behavior of tumor spheroids with and without a BBB, and we had a BBB-only microtissue to also try to understand how the tumor was changing BBB cell behavior. So this, we then add CAR T cells to the devices and observe the CAR T cell migration over time, and also the U87 cell viability, like I showed you in the pictures before. And then those exact same devices were then digested down to single cell suspensions and immediately sent to single run for same-day processing. And so as you can see, the whole process takes about 2 to 3 weeks, which is incredibly fast for a tissue study. Obviously, the data took more time to get back, but that's for discussion. I'm sure that will speed up once we get like maybe AI involved or something. So after getting the data, we had the difficult task of analyzing it all and trying to make sense of it. And here I have to give full credit to our amazing collaborators, Professor Enrico Petretto and Yanis from his team, who have been absolutely vital in making sense of the data. So Yanis first did some standard quality control checks on the data, removing doublets and scaling the data, removing any cells with low quality reads that will interfere with the data analysis. And here I just kind of quickly mention that actually the data from single run was, pretty much, standard. Everything passed QC. Nothing went wrong. You might want to note also that some of the median cell count, the median gene count for U87 BBB CAR T was quite low. And actually, I'll have to mention that like the day that we passed single run that sample, they actually called us back and they were like, hey, the viability is really low. Are you sure you want to proceed? And I said yes, because the viability is low because it's the one that's been treated with the CAR T, so everything is dying. I mean, everything except the BBB cells should be dying. So great customer service from single run there for checking my stuff, but yeah, it's meant to be dying. So after doing all the filtering, you can see here the UAE7 BBB CAR T, we have about maybe only 3.4K cells left after filtering, which was expected. And the median gene per cell was acceptable. So after we're happy with the data, we basically went on to do the cell type annotation. Yeah, so this is kind of important because, especially for our model, we know what cells we put in. So this is a bit of a sanity check for us to make sure that the cells that we put in are the cells that we identify. And this is a tip for anyone who's just starting out in single cell analysis, and because that's where we were. We tried using just one single gene marker to enable the cell types, and that didn't work very well. And what ended up working much better is using a group of cells, a group of genes to label each cell type. That gave us a lot more kind of confidence and robustness with the data. It took us maybe like a couple of months to figure this, couple of weeks to months to figure this out, but giving to you this for free. Here I also want to mention, I think maybe I skimmed it over before, was that the U87 cell line that we generated are overexpressing the tumor antigen marker. So that actually was the only one where we only needed one single cell type gene marker to label the cell type, and that worked really well. And you can see that actually the unsupervised clustering kind of matches also the cell type annotation. So that also kind of just provides us with extra security that we're seeing different cell types and we're labeling them correctly. So once we're happy with the cell type annotation, we can start to kind of look into each condition to identify if different cell states existed within each cell type across the different conditions. And so starting with the simplest condition, which is essentially a U87 tumor spheroid cultured with a BBB, without a BBB in the hydrogel. So it's just a ball of tumor cells in a hydrogel, which is essentially one cell type. We already see some interesting behaviors because despite being essentially genetically identical, we can clearly observe different cell states using the UMAP in the UMAP. And so I've just kind of labeled them here. So the red one is protein metabolism. You've got cells which are more involved in ribosome function and protein synthesis. You've got like a clear cluster involved in cell division and a clear cluster involved in hypoxia, catabolism, and TNF signaling. And if you do the same kind of like analysis with, for example, tumor spheroids, which have which have a BBB co-cultured with them, and you look at either the clusters or and you check which cell types these correspond to, you'll see that in this example, the astrocytes have apparently 2 cell states, while the U87 cells have about 3 cell states. And here I've kind of also just simplified the labeling for you. So we've got one state that's related to hypoxia, one state related to cell division, another cell state that's related also to hypoxia, but also copper and zinc metabolism. So we start realizing that, across different conditions, we see cells in different cell states, and some of these cell states seem to overlap across the conditions and also within each other. And so then we became interested in basically comparing the different cell states for particular cell types across the conditions. So the next thing that Yanis did essentially was to pull out all of the U87 cells from the different conditions, because now we have we're confident of our labeling, our annotation, and then integrating those cells, the U87 cells from the different conditions, such that we could now put them in one data analysis pool and start understanding what are the cell states that emerge for starting with just the U87 cells, like the tumor cells. So here now, what we've done is, what Yanis has done is actually, so essentially integrate the U87 cells across the different conditions and then do a UMAP plot to try and see if we can understand and use unsupervised clustering to see if there are different cell states for the U87 cells alone or with BBB. and also with the CAR T. But let's talk about just UD7 tumor alone versus tumor plus BBB first. And what you can see essentially with this plot is that even though there are fewer cells here, the actual proportion of the cells are very similar. So this suggests to us that the cell states are actually very similar when you go from with or without a BBB, except for those few genes which appear to be maybe increasing the tumorigenicity a little bit. But then what's interesting is that when you then add CAR T cells to the system, so on the top here you have tumor alone, no BBB plus minus CAR T, and on the bottom here is tumor plus BBB, and here is obviously the one that we're most interested in, which is the CAR T cells that have been able to infiltrate the tumor and target the tumor. So you can see obviously there's very few cells because the CAR T has done its job for the most part. And what we're looking here is this is the resistant population here. So these cells are resistant to CAR T therapy. And what you can see is that there's an increase in the proportion of cluster 1 and cluster 5. And also there's an emergence of a new cluster 7, and also potentially 8 when there's no BBB. But cluster 7 emerges only when the CAR T cells are present. So what do these clusters represent? And I'm nearing the end of my talk now. So so essentially, this is almost impossible to read on the slide, but what cluster 1 actually corresponds to is a lot of oxygen response, T cell immune response, and also cytoskeleton organization. So it's a little bit of a mix, but we're still kind of like digging into the actual gene list to understand why they're coming up in together as a cluster. And then cluster 5 is cell division, which is a very kind of intuitive kind of mechanism that you can imagine to overcome therapy, to overcome therapy essentially by dividing more so that they can evade. And then cluster 7 are cells which have increased signaling involved in immune response or cytokine response, which again is completely expected because now there's a CAR T cell there, so they are actually responding to that. But what's interesting is that they're a very small population. So we're trying to now basically, so this is all preliminary data. We're still digging through the gene list and trying to understand how which genes in this set of clusters essentially are likely candidates to kind of target to try to get the CAR T cell therapy to be more effective, and also try to understand why we have a population of UD7 cells at the tumor core who appear to be resistant to CAR T cell therapy. So in summary, we've developed an approach to study the GBM tumor immune microenvironment and CAR T cell therapy in a very controlled way. So because we control which cell types go in, we can actually start to do very clean genetics and and we can do cell-specific genetic manipulations. And so once we generate that list of target genes using the single-cell RNA-seq data, we can then go back to the model, which in theory should be identical to the model in the beginning because we've cultured them ourselves using commercial cell lines. And then we can start to understand if those putative genes are indeed involved in CAR T cell resistance and how they're helping the tumor to basically be more aggressive. And this is an iterative, I already said this. And this is an iterative process because obviously we can then go back and do another round of digest and then check how like any of our manipulations have changed those cell-cell interactions that we predict from the data. And ultimately, we should also be looking towards validating these target genes in mouse models and correlate them with clinical data so that we can hopefully work towards a new therapy. So here is my kind of summary slide where I just want to show that even though I didn't talk about everything that we do, the model basically is compatible with a lot of different kind of technologies, including spatial, which is very, very exciting and novel right now. You know, we really look forward to collaborating with more people on how we can use the model to really dissect the tumor microenvironment. So with that, I just want to thank everybody in my lab. I have 2 labs, and all of our collaborators, and also our funding. So yes, with that, should I'll take any questions. Should I stop the screen share? I don't know, how do we want to do this?
Okay, I think we can stop the screen share, Dr. Maxine. So I do see a question in the Q&A from Nadia. How many sporite culture need to be put for the scRNA analysis? Would that be sporite to sporite variability?
Yeah, so essentially we pooled 3 devices for 1 run, and there is minimal spheroid to spheroid variability. We have other data where we've basically they're essentially technical replicates, right? Each spheroid, because we're not working, this particular data set is not using patient-derived cells or any iPS cells, so everything is essentially homogeneous. Like each cell type is homogeneous in theory, but obviously when you culture them slightly different, operator variability, there will be variability. Yeah. So I think if you check with single run, they need about 20K. I can't remember the number now. They do request for per condition to have about 20K cells.
Okay. Then there's another question. How do you determine the number of genes to use for annotation, and why is it so time consuming?
It's time consuming because you want to get it right. I think it was particularly important for us because we knew what cells we were going in. I think when you work with, for example, patient data or with animal tissues, you may not know what cell types you have. So most people will just use a standard set of gene lists, and then they're kind of happy with it. And they're like, oh, we found all these cell types, yay. But we only have 4 cell types, right? So if we're finding more than 4, there's something wrong. Or if we're not identifying all 4 with good robustness, there's something wrong. So we had to kind of essentially use trial and error to understand which genes it's well known which genes represent astrocytes, parasites, and everything, but it was there was a bit of experimentation to understand which genes were the best at labeling.
Okay. Thank you for the answering the question. There's another question in the chat box. It's from Serum. Great talk. Are there any GBM mouse model existing right now to mimic pathology manifestation of the clinical disease, or would this be tested on mouse xenograft models?
Yeah, so I think essentially the current state of the art is GBM mouse models, and they do manifest a lot of the pathology that you see in patients, but obviously there are issues with species compatibility, especially when you work with T cells. So that's why there was this a need for kind of a human-based, like a model that only uses human cells essentially for T cell therapy. But also, and yes, the idea is to eventually compare what we find with a mouse model, which is actually something that we're also doing for a slightly different project. And we do see a lot of similarities between what we see and what we see in the mouse, yeah.
Okay, so there's another question. Any advice for labs that are venturing into single cell sequencing and data analysis for the first time?
I think make sure that you identify the right company to work with. There's obviously multiple companies that can do single cell sequencing, and I would say from the bioinformatics point of view, like there's no like you get a gene matrix, right? And so it's just which platform you feel is most convenient for your setup. So I'm doing a bit of publicity for a single run today, but we personally like had a very good experience working a single run, and we felt that their prices were fair. Yeah. So yeah, so I think that's the advice. Like, make sure that you find the right company to work with, and I would definitely try to also find bioinformatician to work with you rather than figure this out on your own.